Welcome to online coaching program of SAP UAE Medical Microbiology Technologist. Today we are discussing on mycology. Mycology is the study of fungus. It is an important area coming under microbiology technologist examination. So first of all, we can discuss about the what do you mean by mycology. It is the study of fungus, and you know that fungus is an eukaryote and majority of the organelles are present in this fungus, and the cell wall is made up of chitin, cellulose, mannose and other polysaccharide. And when talking about its cell membrane contains sterol. And when we talk about the reproduction mode, sexual and asexual mode of reproduction is present. And while discussing about the different forms of the fungus, simplest fungus is the unicellular yeast. Multicellular forms have tubular filamentous structure called hyphae. Two type of hyphae is mainly present, septate and aseptate hyphae. Aseptate hyphae is also known as cenocytic hyphae. And this hyphae together they constitute mycelium. That is, mass of hyphae together form mycelium. It is about the basic introduction about the fungus. Next, about the classification of fungus based on morphology. There are mainly four types of fungus: yeast-like fungi, mold or filamentous fungi, and also dimorphic fungi. So first we discuss about the yeast. Yeast is the unicellular spherical shaped fungus, that is called yeast. Unicellular small organism spherical shaped fungus is the yeast. They mainly reproduce by budding, and on culture they form smooth creamy colonies, and examples for yeast include Cryptococcus neoformans. It is a capsulated fungus and it is the main causative agent of meningitis. Meningitis is an important inflammatory infection that is appearing or involves meninges. Next type is yeast-like fungi. They grow partly as yeast and partly as filamentous or mold form. They can exist in yeast form and mold form. Example is Candida albicans. Next type is mold or filamentous fungi. The fungus which produces true mycelium are called mold form. They are filamentous forms of fungi. Can produce different types of spores or conidia. Major type of spores are produced by this mold or filamentous fungi. Example include dermatophytes. Next type is dimorphic fungus. They exist in filamentous or yeast form depending upon the condition of growth. In different condition, they exist either as yeast or either as mold form. So, in host tissue, that is in the body itself, that is culture at 37 degree, in the body temperature, they exist as yeast. The same organism or same fungi when present in soil and at culture at 22 degrees Celsius, that exists as mold. So this dimorphic fungus partly exists as yeast, and sometimes it exists as yeast and sometimes it exists as mold depending upon the condition for growth. Example include pathogenic fungi that cause systemic mycosis. Fungal infection that affect internal organs or affect the organs are called systemic mycosis. Next part include the lab diagnosis of fungal infection. So when you're talking about the sample taken for this laboratory diagnosis, the main samples include skin, nail and hair. For first technique is KOH mounting. So in this technique, 10 to 20% of KOH is added to a sample taken on a slide, and it is observed under the microscope. So it is an important technique for determining fungal morphology. This why we should use this KOH is that KOH readily dissolve the keratin in the clinical material that is present in the nail, mainly present in the nail. The keratin material is dissolved in this KOH. Fungus remain intact due to the chitin in the cell wall. So for snail specimen, 40% of solution of DMS, that is dimethyl sulfoxide, is also added to KOH in order to obtain the nail specimen. Next is lactophenol cotton blue staining. This lactophenol cotton blue staining is an important method of staining used in fungal staining procedure. In this lactophenol cotton blue, the lactic acid is used to preserve fungal morphology. Phenol is used as a disinfectant or for killing organism. Cotton blue is used as a dye or stain. Glycerol prevent drying of the smear. Next is calcofluor white stain or blanco-for white stain. It is a fluorescent whitener used in textile industry. Here 15% KOH and calcofluor white solution is added. So after placing this 15% KOH and calcofluor white solution, place the material to be examined on the clean glass slide, and after it drop of 15% KOH and calcofluor white stain is added, and place a cover slip or cover glass above it and examine under fluorescent microscope. Calcofluor white stain plus Evans blue will give a apple green fluorescent to fungi. Calcofluor white stain will bind to cellulose and chitin of the fungal cell wall so that we can obtain a clear image. Next is periodic acid shift stain. It is used to differentiate actinomycetes and fungus. So what is the difference between actinomycetes? Actinomycetes is a gram-positive filamentous bacteria and it is like fungus. It is filamentous like fungus, but it is bacteria. So in order to differentiate between actinomycetes and fungus, we use periodic acid shift stain. It stains polysaccharide in the cell wall of fungus, and the fungus stain red, magenta, pink red color with blue nuclei. Actinomycetes are not stained by periodic acid shift stain. Next is Grocots gomoris methenamine silver stain. Silver nitrate outlined the fungi in black due to silver precipitating on the fungal cell wall. So fungus will appear black in color. It only outlined the fungus structure. Next is Giemsa stain. Yeast form of Histoplasma capsulatum can be stained by this Giemsa stain. Next, different culture media used for the cultivation of fungus. First one is Sapporo-dextrose agar. You are familiar with this fungal culture media. It is the most common media used in microbiology laboratory, and its pH is 5.4 to 5.6. Histoplasma capsulatum and some strains of Nocardia asteroids do not grow in this media. This organism does not grow in this medium. Next is modified SDA. SDS plus cyclohexamide plus chloramphenicol is used in this, used mainly for the isolation of dermatophyte, mainly for skin, nail and hair specimen. Next is brain hurt infusion agar. It supports the growth of bacteria and fungus including histoplasma. So it is not used for skin, hair and nail specimen. Other specimen we can use this for both bacteria and fungus. Next is modified BHIA. BHIA കൂടെ cyclohexamide chloramphenicol കൂടെ add ചെയ്തിട്ടാണ് modified BHIA prepare ചെയ്യുന്നത്. Dermatophytes ഗ്രോത്തിനാണ് മെയിൻ ആയിട്ട് യൂസ് ചെയ്യുന്നത്. Next is cornmeal tween 18 agar. It is used to demonstrate blastoconidia pseudohyphae. Pseudohyphae എന്ന് പറഞ്ഞാൽ മെയിൻ ആയിട്ട് Candida albicans കാണുന്ന ഒരു സ്ട്രക്ചർ ആണ് pseudohyphae. Arthrochonidia chlamydospore produced by Candida species and other yeast. Next is potato dextrose agar, used to enhance sporulation and pigmentation, also used as a subculture medium. Next, inhibitory mold agar, used to require pathogenic fungus except dermatophyte. Mycosal agar, used to recover dermatophyte. ബാക്കിയുള്ള species dermatophyte മാത്രം recover ചെയ്യാനാണ് mycosal agar-ഉം അതുപോലെ dermatophyte അല്ലാത്ത pathogenic fungus-നെ recover ചെയ്യാണ് inhibitory mold agar യൂസ് ചെയ്യുന്നത്. Dermatophyte test medium used for the isolation of dermatophyte. Bird seed agar used for the growth of Cryptococcus neoformans. Next, trichophyton test agar used to grow trichophyton species. Germ tube media used to demonstrate germ tube. This germ tube is a important characteristic of Candida albicans and Candida dupliensis. So germ tube or Reynolds broad phenomenon. It is otherwise known as Reynolds broad phenomenon. So it is used for the recovering or demonstrating germ tube produced by Candida albicans. Next is urea agar slan, used to detect urease production of fungus. Positive test pinkish color കാണിക്കും 48 hours incubation കഴിഞ്ഞാൽ. Urease positive ആയിട്ടുള്ള fungus ആണ് Cryptococcusum trichosporon. Urease negative ആയിട്ട് most Candida species അതുപോലെ Saccharomyces geotrichum ഒക്കെ urease negative ആണ്. Urease test used to differentiate Candida and Cryptococcus. Next is gram stain. Fungus gram positive ആയിട്ട് അല്ലെങ്കിൽ blue കളറിലാണ് appear ചെയ്യുക. അപ്പോൾ നമുക്ക് ഈസ്റ്റിനെ അതുപോലെ സ്യൂഡോ ഹൈഫനെ ഒബ്സർവ് ചെയ്യാൻ നമ്മൾ എന്ത് ചെയ്യും? ക്ലിനിക്കൽ സ്പെസിമെനെ ഒബ്സർവ് ചെയ്യാൻ നമ്മൾ ഗ്രാം സ്റ്റെയിൻ യൂസ് ചെയ്യും.